Hello, everyone, and welcome to Simultaneous Detection of Respiratory Pathogens using Microfluidic Singleplex qPCR for Surveillance. I'm Cassie Soltman of Labroots, and I'll be your MC for today's event. Today's educational web seminar is presented by Labroots and brought to you by Standard BioTools. To learn more about our sponsor, please visit standardbio.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I do also want to mention that there will be an interactive survey that will populate after the presentations conclude, and we appreciate your participation. I'd like to now welcome our speakers for today's webinar in the order in which they'll be speaking. Jeff Dance, Senior Field Application Scientist, Team Leader at Standard BioTools. Thomas Saville, Technician at the University of Sheffield. Brad Meyer, Senior Field Application Scientist at Standard BioTools. And Dr. Amy Ellis, Senior Scientist Level 3 at the University of Wisconsin-Madison. Jeff, I will now pass it over to you to begin the presentations.
Thank you. Good afternoon, good morning to everyone. Welcome to what promises to be a great webinar on respiratory pathogens. So I'd like to give a quick introduction to Standard BioTools before we get to the main presentations. We're Standard BioTools, formerly known as Fluidigm. We're a global company with over 20 years of experience developing innovative products with patented technologies that enable various genomic and proteomic applications. We're headquartered in South San Francisco, California, and we have research and manufacturing facilities in the US, in Canada, and Singapore. We have approximately 600 employees worldwide, and our genomics products have been used in over 9,000 publications to date. Standard BioTools has a broad portfolio of products in both the proteomics and genomics fields. We have instruments for flow cytometry and spatial biology that are not limited by issues of fluorescence deconvolution and background. And we recently merged with SomaLogic to be able to offer protein detection and measurement at a vast scale. If you or your colleagues have interests in proteomics, please reach out for further information. However, today we are focusing on our microfluidics genomics platform. So the microfluidic integrated circuit, or IFC, which is shown here on the very right, was developed by Fluidigm almost 20 years ago and continues today in conjunction with the Biomark X9. And the X9 itself is a development of previous generations of Biomark instruments. The Biomark system is highly versatile, as can be seen here by the range of key applications, and the numbers in the circles represent the number of peer-reviewed publications for each key area. Today, we focus on pathogen detection, and we're going to hear about some recent studies in pathogen research from two recently published scientists. In both cases, they used a high-throughput method of detection based on qPCR and the Standard BioTools Biomark platform. I'll not give away any details of the studies, but I'd like to spend a few minutes giving some background on the technology that helped with their research, specifically the Biomark platform and the integrated fluidic circuits. So Biomark can offer significant benefits in microbial pathogen studies, surveillance, early detection, root cause, and the potential for early intervention. And we have customers across the world investigating respiratory disease, emerging pathogens, and antimicrobial resistance, gastrointestinal disease, and other opportunistic pathogens. This can be in human health, wastewater surveillance, food safety, animal health, and fisheries, to name but a few. So our core technology is delivered to our customers through a consumable that has the SBS microplate form factor, the IFC or chip, as people call it. The consumable is run on a system we call the Biomark X9 that has the capability to move fluids, thermal cycle, and perform optical detection. And the overall workflow looks like this. So the chip has wells where the user can pipette samples and assays. After loading the chip with the samples and assays and reagents provided by our kit, the user would place the chip onto the X9 system or a similar older device. And with the single push of a button, the system is able to mix samples and assays, carry out the reactions, make the optical measurements, and report analyzed data in a walkaway workflow. As a point of reference, we utilize integrated fluidic circuits, which automate the assembly of PCR reactions. Now, most people are familiar with 96 or 384 well plate setups, where you add all components of a PCR mix to each individual well. The IFC only requires that you add diluted assays into dedicated assay inlets, shown on the right here, which are separate from the samples, which are mixed with a master mix and also pipetted onto the IFC. By adding reagents separately, we allow the IFC and biomark to automate the reaction assembly, which ultimately uses much less reagents and reduces errors significantly when compared to completing the process manually with an 8-channel pipette and 96 well plates. So the microfluidic system moves the assays to all the nanoliter volume reaction chambers in the chip, and then the samples are moved into these locations. And it's important to note that all the samples see all the assays, but the assays are not mixed. These are singleplex reactions. And in this way, we can generate up to 9,216 individual PCRs on a single chip. So let me just present a hypothetical project to you as an illustration of the power of this method. Let's say we have a project with 1,000 samples and we have 96 targets of interest, be they SNPs or cDNAs. To achieve this by traditional PCR in a 384 plate format would require 250 runs. And if we assume one technician, one PCR machine, 3 runs per day and 5 days per week, it would take almost 4.5 months to achieve this. Let's consider the same project on the Biomark X9. We would require only 11 IFC runs, which could be achieved in 4 days, or let's say 11 days if you want to relax into it. Imagine what you could do with the remaining 4 months. There's also a significant saving in PCR master mix, and indeed in the number of tips you'll consume. The final part of our offer is our D3 assay design group. We have a team of molecular biologists dedicated to optimizing the primers you'll need to achieve this scale of work. You can simply upload the target details via our portal, and we will design optimized assays for you and pay attention to minimizing off-target interactions. And by way of example, I've listed the current tally of pathogen species that we've designed for that are in the public domain. Or, of course, you can use your own assays. That's entirely flexible on how you would choose to achieve this. So to summarize, let's talk about the real-world benefits of our technology. Cost efficiency, we're using tiny volumes of reagents in a nanoliter scale reactions, which means big savings on consumables. We have high capacity, so we can complete up to 46,000 PCR reactions in an 8-hour shift if you run multiple IFCs in a day, or we can craft up to 384 barcoded libraries every day. And I'll mention we haven't even touched on our workflow for making amplicon-based next-gen sequencing libraries. That's perhaps for a different webinar. And finally, we have some simplicity and range of application. So even with this high capacity, the system keeps it simple. You can detect up to 96 targets at once without complicating the workflow. So in essence, the biomark is designed to do more, cost less, and stay easy to use. It's all about getting you the data you need quickly, efficiently, and on a large scale. So that brings my overview to a close. I hope you found it informative. So let's get started. Our first speaker today is Tom Saville from the University of Sheffield. He's published papers on respiratory viruses and will present some recent findings today. And with that, I'd like to hand over to Tom.
Hi, thank you, Jeff. I'll just hopefully the slides are moving. There we go. Yeah, so as Jeff said, I'm Tom Saville. I work at the University of Sheffield, and today I'm going to talk about microfluidics and detection of common respiratory viruses, specifically in children with influenza-like illness, from this study that I recently published. And so the overview of my presentation: respiratory virus incidence, how important is this? Current and established methods of detection, assay development and how we transferred the assay to use microfluidics and Fluidigm's platform, the assay application and the results, and the implications of this type of microfluidic assay for future surveillance, specifically, again, respiratory viruses. So the global incidence of common respiratory viruses has maybe been on people's minds a lot recently since SARS-CoV-2 emerged, and since the emergence of SARS-CoV-2, there's been a cumulative 7 million, over 7 million excess deaths. The other common respiratory pathogens you'll probably be highly aware of are influenza, RSV, which has a lot of vaccine development work going on recently, well, has been approved recently, and then others such as adenovirus, which can be highly, well, a big problem in children globally and in low to middle income countries. Rhinovirus, your common cold, human parainfluenza virus. So the instances of these generally quite high, with varying hospitalization rates and deaths, variable by countries and the country's income. A lot of these stats taken from the World Health Organization website, although there's some cases where the virus isn't necessarily tracked or sort of monitored as well as other viruses. For example, influenza and SARS-CoV-2, very well set up for global monitoring. Rhinovirus, very difficult because it's such a common cold, which often doesn't result in hospitalization. But yeah, there's a lot more viruses than just influenza and SARS-CoV-2. So the symptoms of a lot of respiratory viruses are very, very similar, and despite handy charts as shown here, it's often impossible to pick out what you've got from deciding if you've got a fever, a cough, or a sore throat. They're so generic, and the only real way to tell is by a molecular diagnostic method. You could say well, it probably still is the gold standard molecular diagnostic method for respiratory virus detection is taking your sample, generally a nasal swab, and extracting your sample, reverse transcribing, so converting that RNA into double-stranded DNA, and going through various PCRs to amplify that fragment of interest so it's detectable. And we detect this via a probe, fluorescent probe, in a standard qPCR assay. So the point at which, I suppose, the fluorescence is detectable above the threshold on this PCR machine is the point at which we say it's a sort of, well, it's after the number of cycles. So the higher the cycles, the less viral load is in that sample. If you just want in a standard assay to say yes or no, yes, if it fluoresces, then it's there, but it can be quantified via the curve and point at which it fluoresces. So pretty standard qPCR. And these qPCR type assays are used clinically, quite common clinically, especially in the UK and the NHS. And there'll be a set viral panel, which is used to determine if there is a pathogen in a clinical sample. We actually took the current established NHS test for our local area within Sheffield, which consisted of the viruses shown here on the left. And we wanted to adapt that and update it for use in research, but also for use on samples within a recent study in the Gambia. So it needed updating as it didn't include SARS-CoV-2, but including bocavirus and another human coronavirus, HKU1, would also be of interest to see what is actually circulating in the population we wanted to test. You could have swapped these out for any viruses you're particularly interested in. If you wanted to be more interested in influenza, you could separate your probes into different strains of influenza, although it would take up, you'd just end up with a lot more targets in the end. So yeah, the goal was just to update this, as it didn't include SARS, didn't include another common or fairly common coronavirus, and human Boca virus, which we were interested in the population. And this increased the number of targets, and we realized that actually this would be quite a big job to multiplex all these targets, and that's if we could multiplex 4 probes and primer probe sets per well. The NHS test is a multiplex test. Not all the viruses were 4-plex, some were 2-plex due to primer probe interactions between sets. And what also helped their setup is that they've got a semi-automated system, and we were going to do this by hand. So it meant a minimum of 5 multiplex reactions of 4 primer probe sets, which seemed quite daunting. And it would have taken up a lot of PCR plates and a lot of time. As Jeff mentioned, you can drastically reduce the plasticware and the time it takes to go through all these tests by using microfluidics. And we had the biomark available in the university to use. So the idea was actually we might as well make use of this machine we've got and try and transfer this assay over to a microfluidic sort of workflow, considering the number of targets we had and the number of samples we wanted to analyze. As Jeff also mentioned, it means that we don't have to run these reactions in multiplex. Each reaction is a singleplex reaction, so it made also ordering all the primer probe sets quite easy because we could order them on the same color, just standard FAM fluorophores. And we were less at risk of having primer and probe interactions between sets. So primer dimers form in between, say, like an influenza set and an RSV set, which we would have just taken a lot of time to figure out if that sort of happened, and could also result in false positives for either virus. So it just avoids that whole sort of interaction. So I thought it was a good idea, and went about developing, looking at how we could use this technology. So you still have to go through the extraction phase and reverse transcription phase. The difference compared to a standard 96-well plate qPCR is that we have to do a pre-amplification phase. Just because you're loading so little product into the final plate, I think 3 microliters of each sample and 3 microliters of probe for the whole plate of reactions. So there was already a SARS-CoV-2, luckily, protocol by Standard BioTools, and that had a pre-amplification step within it. And I can't remember if we actually just took the same number of cycles, but we did do a small assay of determining how many cycles the pre-amplification step should be. And we landed on 20 pre-amplification cycles. You pre-amplify using the primers. We did this without the probe, just because we ordered custom primer probe, so we could afford to just not put the probe in. If you're using standard off-the-shelf primer probe sets, then you can leave the probe in, but it's sort of a bit of a waste because it's not being read at this point. It's just PCRing up your target, so you've got a bit more of it to load into the plate, into the IFC. Load the IFC, and then we effectively do the qPCR on the biomark, which looks a bit different to the biomark that Jeff showed, because I think as an older model, and we had to put our plate on a machine to load the plate, and then on the biomark to read the plate. And it sounds like the new biomark loads and reads at the same time, but I can't confirm this, as I don't know the products inside out. So once you've done the amplification and loaded it into the plate, which can be multi-channeled, we can run 192 samples in this case against 24 primer probe sets, just by loading the samples and the probes and letting the machine and the plate do the rest. So we decided it was best to validate this assay before we just went and used it on our our research samples. So we managed to get hold of samples from Sheffield teaching hospitals that had undergone qPCR via their sort of standard in-house qPCR test. And we correlated our positives with their positives to basically see if our test was reliable. Does it detect what we expect it to detect? Does it detect positives? Is there dropout on any particular primer probe sets? And actually, it was quite difficult because although we got a lot of positive samples sent over for validation, trying to get enough positive samples from each virus to have enough power to deduce if it was working or not was quite difficult. I think we would have to run this alongside for many more months. But the important ones that we managed to sort of validate by a sort of what do you call it, correlation were SARS-CoV-2, RDRP, E. They had enough samples for RSV, rhinovirus, and you can see the adenovirus in the top right-hand corner is one where we barely had enough samples to draw a reliable sort of line of regression. I think it did show that the challenges are the CT values don't perfectly correlate. So if you had a CT of 30 in a standard qPCR, it wouldn't be 30 in the microfluidic PCR. And to combat that, as I said, we could change the pre-amplification cycles, but we didn't want the CTs to go too low or too high, particularly. So when we settled on 20, it was probably about right. We weren't getting many dropouts as non-concordant in each case. We also did ROC curves and a sensitivity assay with known positive controls, which is any additional supplementary data of the paper, but it would be a lot to go through for each virus for this presentation. But ultimately, we were happy with what we were picking up compared to the clinical samples from Sheffield. So we moved on to applying this assay to our sort of cohort, which was in the Gambia. So this was residual samples from a upper respiratory study called NASImmune. So the NASImmune study was the trial of a live attenuated influenza vaccine in healthy children in the Gambia up to, well, between 24 and 59 months old with no underlying illness. And when they were, different groups were either given the vaccine or the placebo, and they were enrolled to follow-up appointments throughout the 2 years after that. So they would, if they presented with symptoms of fever, cough, or a sore throat, and then they would be called to clinic, and swabs would be taken to see effectively what viruses they've got. I think initially it was to detect if they had flu, but we wanted to detect if, I suppose, as the symptoms are so general, if what they were carrying, and also it gives an idea of where what we should do next with effectively studying low to middle income countries virus transmission, and a lot of the people in our respiratory infection research group are looking at various viruses and also bacteria within that cohort, as we have a lab there. So it'd be just interesting to see what is circulating and if there is as much flu or is it other viruses like Oca virus that we started to insert into our panel. So we had 158 residual samples to test, which is lower than expected, but that's what we had available in the lab. And we decided to test these in duplicate, just so we could rely on the results effectively. We did have a few cases where it was useful to have it in duplicate, because we found that a few samples did have just sort of drop out, and I don't know if that was due to loading issues or amplification issues, or that the samples were actually at this point quite old and been frozen for quite a long time for RNA. And we ended up with 121 positives and 37 negatives. I mean, we're expecting a high number of positives because these children are presenting with a respiratory illness. The highest instance was rhinovirus, which was expected, your common cold. And we also detected quite a high level of adenovirus, which we thought was quite interesting. And it is not, I don't think it was commonly reported that adenovirus is circulating that much in West Africa. But I think maybe more research is needed into how much adenovirus is circulating, and also what strains, because it's very strain dependent on how severe adenovirus can be. Lower than expected influenza detection, but this is probably due to a lot of these children being vaccinated against influenza. So that is expected. It's not a true reading of incidence of influenza within this cohort, within that country, because of where the samples were taken from in this study. And we detected no SARS-CoV-2. We included this to work up the panel and potentially use it as a clinical tool and a research tool. In this cohort, it's good that we didn't detect any SARS-CoV-2, because it shouldn't have been around in 2017, 2018. And if we did detect it, it would have shown that either the assay is producing false positives, or SARS-CoV-2 is more abundant than we thought it was. So that was good news. A few of the interesting insights we got from this test was co-infection. So a third of the positives had multiple viruses detected. So there was a lot of co-infection, generally between adenovirus, rhinovirus, so your common cold. And as I said, adenovirus, quite interesting in that it's not commonly reported. And that adenovirus took up 79% of the adenovirus cases were actually co-infection cases with other viruses. So again, it's an interesting sort of area for further research, how adenovirus is circulating in West Africa, and if it is actually symptomatic, or if it's the other co-infection that is producing the symptoms, and it's more of a carried infection. Seasonality. So with the data, we could plot seasonality of the virus, and it was nice to see that most of the time, year-on-year seasonality matched up fairly well, and how, especially with I think which one was it? The green one here. I'm sorry, I've not got it big enough on my screen. Parainfluenza, how it can flip-flop between one or two or different parainfluenzas depending on the year. So if we only managed to look at 2 years, but obviously in 2017, one was more prevalent than the other. And the Gambia is quite interesting in that it has a predominantly dry season, a predominantly rainy season, not your classic winter, spring, summer seasons. So the dry and wet season, there was a clear difference in which viruses circulated when, and especially if you look at the lower chart, which is just the negatives. You had generally more negatives towards the end of the dry season, and so more positives going into the rainy season. which again, we sort of expected with respiratory viruses just because of the humidity of the rainy season. And also a lot of people tend to spend more time indoors in the rainy season. So it's nice that we could make these links. Similar to the NHS Sheffield Children's Hospital validation samples, we didn't actually get that many samples for each virus that were positive. And you'd need to sample so many more children over a much, I suppose, longer period to get the number of samples required, to get the number of positives for each virus to give it enough power and to draw more meaningful conclusions about seasonality and potentially even co-infection. But it shows that we can actually use this technique and implement it to produce data like this. So it's potentially useful for future studies or planning future studies where we actually plan microfluidic and viral detection into the study rather than just doing it after samples have been collected as a project, sort of additional project later on. So for future respiratory surveillance, I suppose this is very interesting post COVID-19 in that sort of pre-COVID, the World Health Organization was mainly focused on flu, and thus we have the Global Influenza Surveillance Response System, tracking flu and flu strains globally. And it was proven through the pandemic that this can be successfully adapted to monitor SARS-CoV-2 and also other epidemics like the MERS-CoV epidemic. I think given the technology and how we can detect multiple viruses in single tests, it'd be nice to see a broader respiratory virus surveillance platform and program which could interlink different viruses. So basically adapting the current system to include RSV, to include human metapneumovirus. So the data can be uploaded, and with a system like this where it's many primer probe targets from one sample and one swab, you can look at co-infection, and I think globally it would greatly enhance our pandemic preparedness, because we'd have data from how these viruses act year on year, and if certain viruses co-infect with other viruses. It just seems that the monitoring system is still predominantly focused on influenza, whereas it could be expanded to more viruses, and it would aid research and therapeutic interventions, vaccines, stuff like that. And also I've put next strain there because next strain did a very good job of strain tracking, especially during the COVID pandemic. And although this is just PCR detection, it can feed into sequencing. Once you've got a positive on a sample, there's no reason why that can't be fed into a sequencing pipeline, and then you can gather data on strain, more detailed data on strain than if you just had a qPCR panel that managed to resolve the strain. So I think sequencing is sort of a next step of surveillance, is definitely something that these sort of systems could feed into. How I see this assay being implemented is maybe respiratory surveillance of say in the UK of inpatients, or not necessarily inpatients all the time, but what am I looking for? The severely ill patients, so ICU patients as well, and maybe being tested for specific viruses in your local lab like Sheffield does. But this sort of setup requires a lot of samples. It requires a high throughput of samples to make it cost efficient. You don't want to be waiting for samples to fill a plate. So it works better as like a centralized lab system. So either a regional lab where samples can be pulled from multiple locations, or a national lab depending on the size of the country. So for example, if the study was going to be set up in the Gambia, it would probably just be a national lab in the main capital or in the medical research unit, where swabs can be pulled from all around the country and tested, potentially weekly if enough swabs are collected on a weekly basis. But what you don't want is your RNA and your swabs sitting in freezers for weeks and months, because it's going to take you a while to collect your data, and also they can be degrading. So it's always best to try and test these types of samples as soon as you can, batch them together. And it is quite easy to swap to new primer probe sets. So should another pandemic come along, you can just swap in new primer probe sets, quite quickly validate those, and adapt the tests based on your country, your region, local outbreaks, bird flu potentially, stuff like that. So as long as it's coming from that same swab, or a single sample, you can multiplex. Well, it's not multiplex. You can add whatever primer probe sets you want up to the limit of the plate. In our case, 24, because we used the 19224 plate. So I suppose that concludes my talk, and I might end up, I suppose, provoking more questions than it answers, but hopefully it gives a good overview of how microfluidics can be used for viral surveillance in different settings, and potentially replace a standard qPCR should sample throughput be high enough. And this paper, all the data I presented here is published already within the paper listed on this slide, Microfluidic qPCR for detection of 21 common respiratory viruses in children with influenza-like illness. And I'd like to thank Professor Tushan De Silva, who is my PI, and Dr. Haley Colton, who was the co-author of this paper with me, and has a lot more clinical knowledge that she brought to the paper than along with my technical knowledge. So thank you for listening, and if you want to get in touch, my email is there, or if you've got any questions, please just post them in the chat if you found the Q&A box. And yeah, thank you. I'll pass on to I think it's Brad next.
Hi, yes, thank you, Tom. That was great talk, and I'm going to move on to the next presenter. Next up is Dr. Amy Ellis. Dr. Ellis is a senior scientist in the laboratory of Dr. Shelby O'Connor. She received her PhD in Dr. Janet Mertz's lab at the University of Wisconsin-Madison in the cancer biology program, and did postdoctorate research studying Ebola and influenza in Dr. Yoshi Kawoka's lab before joining the O'Connor lab. The O'Connor lab studies immunopathology associated with disease such as Mycobacterium tuberculosis and HIV, but has also recently begun studies in environmental surveillance of respiratory viruses. Their specific interests are using air samplers to monitor respiratory viral pathogens in the community, but they are currently expanding to global sites as well. And without further ado, I'll hand it over to Dr. Amy Ellis. Take it away, Dr. Ellis.
All right, thanks, Brad, for the introduction. I'm Amy, and I'm in Shelby O'Connor's lab at the University of Wisconsin-Madison. And today I'll be talking about the work we've done with detection of multiple respiratory pathogens in air samples using the Standard BioTools custom respiratory pathogen assay panel. To give you an outline of my talk, first I'll give a bit of background about the air surveillance program we have in our current K-12 school study. Then I'll describe the current method, multi-pathogen panel that we're using for air sampling. It's got quite a long name for the purposes of this talk. I'll be calling it the Carmen assay. And I will also describe some of the limitations we've found with using the Carmen assay on air samples, which leads me to the results where we tested the custom Standard BioTools probe-based multi-pathogen panel assay with air samples and compared that to the Carmen and quantitative reverse transcription PCR results. Finally, I'll give you some conclusions and future directions. To give you an outline of our K-12 air surveillance program, Currently, we're performing a K-12 study. This has been ongoing for a few years now. Our goal for this study is public education of community spread of viruses. So within the scope of this project, we collect air each week using Thermo Fisher's aerosol sense air samplers, which is shown in the picture on the right. And we have these air samplers placed in 17 schools in the Dane County, Wisconsin area. So the cartridges for these air samplers are changed out every 3 to 4 days and then delivered to the lab for processing. And we share our data weekly to public health dashboards. On this slide, I'm showing you the respiratory illness dashboard. We've partnered with the Public Health of Madison and Dane County, and they show the data here shows that from the air samplers that are placed in these schools, the percent of positive samples each week in either elementary or middle and high schools. So now I'm going to get into a little bit more of the sample processing pipeline we use. So we run those air samplers, again, 3 to 4 days typically for each sample. Then we remove the cartridges that are inside the air sampler. And within each cartridge is a couple of foam substrates. They're kind of squishy material. And we squish those down into tubes that have PBS to elute any genetic material that's present on the foam substrate. We then isolate and isolate and extract the nucleic acids. And then we do downstream assay testing. So the different assays that we've used, I'm going to show 3 different examples of ones that we've done for these air samples. Currently, we're using multi-pathogen PCR assay detection, but we've also we've performed metagenomics on these samples, as well as when we first began, we were doing quantitative PCR, and now we're moving into digital PCR assays as well. So why should we use multi-pathogen PCR assays for air samples? Typically, the volume that we get after nucleic acid extraction is extremely limited. So we get about 50 to 100 microliters per sample. There's also not a lot of genetic material collected on the air sampler. So we typically have to figure out what to do with it in a realistic fashion. So the more sensitive and quantitative detection, like for qPCR or digital PCR, currently those platforms don't support surveilling a large number of targets. Most of the platforms, typically you can look at 1 to 4 targets. Also, the cost associated with doing digital PCR on a large number of targets, and also for qPCR, is much higher than that is what's associated with the multi-pathogen panel detection. Finally, we do have higher sample throughput, which I'll get into on the following slides for multi-pathogen PCR assays. Typically, we can run 48 up to as much as 192 samples on some of these platforms. The current multi-pathogen panel assay that we're using is the Carmen assay, and it's a CRISPR-Cas13 based PCR assay that was developed by Pardis Sabati's lab at the Broad Institute in Boston. They developed this during the COVID-19 pandemic to use as high-throughput screening of clinical nasal swabs for patients with respiratory infections. We adapted the Carmen assay for air samples in the 2023-2024 school year, which we're currently preparing a manuscript for this. To give you an example of what the pipeline looks for the Carmen assay, just to have a comparison for the RPP assay later. We take nucleic acids and then we perform reverse transcription PCR with multiplex primers. And one important note about these primers is that they have a T7 promoter overhang on the 5' end of the forward primer. So all of the template DNA that we make has a T7 promoter region. And that's important for the next step, which is performed on the Biomark X9, and it's run on a 19224 IFC plate. That means that we can run up to 192 samples with 24 assays. So in that assay, we have our template DNA with the T7 promoter, and the RNA polymerase then can bind to and produce target RNA. The target RNA then is recognized by the CRISPR RNA of interest that's present in our assays. And then the Cas13 enzyme can bind to and cleave that target RNA. there's a poly-U reporter probe that has FAM fluorescence attached to it that, as part of a collateral cleavage mechanism of the Cas13 enzyme, produces FAM fluorescence, which is read in individual microchambers in the middle of that IFC plate. Currently, there's 15 targets in the Carmen assay, and they're shown here on the right. So we have 2 targets for SARS-CoV-2, as well as seasonal coronaviruses, metapneumovirus, human parainfluenza virus 3, RSV. We have flu A and flu B targets, both the PB1 gene, and we expanded it for air samples to the M gene as well to get a greater sequence diversity, particularly for flu A. We also looked at adenovirus, and we have a human control, which is RNase-P. For the 2023-24 school year, we compared Carmen to quantitative PCR. And over here on the right, I'm showing you the results for SARS-CoV-2. qPCR shown in the black lines. Over time, our sampling date range from September 2023 to June of 2024. And I'm showing you the percent of positive cartridges on the y-axis. You can see for SARS-CoV-2, both qPCR and Carmen data follow that same trend line where there's an increasing amount of detection of positive cartridges. This is kind of the winter months. Currently, SARS-CoV-2 doesn't seem to have a season, but we did observe more of it in the winter for this school year. However, we did not observe any concordance between qPCR and Carmen for flu A. And this was even after expansion, where we tried to increase the Carmen sensitivity by including the M gene target. So overall, there's some limitations to the Carmen assay. Its advantages is that it does have very specific detection of targets. However, with air samples, if those targets are degraded, we can often miss detection of some of those samples. The disadvantage is that there is a pre-amplification step with the T7 overhangs. Those long primers are somewhat inefficient at that producing DNA. The Cas13 enzyme, I'm not going to get into this today, but it's highly sensitive to PCR inhibitors that are prevalent in the air samples. Dirt and some cleaning products have gotten into the air samplers, and it completely knocks out the ability of that Cas13 enzyme to function. Finally, it's difficult to design CRISPR RNAs for viruses that have really high sequence diversity. We see that with influenza A, but we've also seen that for a few other viruses, particularly like rhinovirus and enterovirus. I don't include those on the Carmen panel because they're so sequence diverse. We don't have very good sensitivity. So we've been exploring alternative multi-pathogen detection panels for air samples, which leads me into talking about the Standard BioTools Custom Respiratory Pathogen Panel, or RPP assay. The workflow for this assay is shown below. So we have our nucleic acids. I'm showing RNA here, but there's DNA and RNA both in that tube. We perform a pre-amplification step, RT-PCR step, where we amplify any DNA present in the sample. And these are standard primers, so the amplification step is a little bit more efficient compared to the Carmen. Using that pre-amplified DNA, then we load onto a 48 by 48 chip on the Biomark X9. And again, all of the reactions occur in these individual microchambers in the center. In this case, FAM fluorescence is probe-based, and it's measured every PCR cycle. So we get a CT value readout for the RPP assay. The targets are very similar to the Carmen. There's 17 of them with room for expansion up to 48. We have SARS-CoV-2 as well as seasonal coronaviruses and influenza A and B viruses as well. Human metapneumovirus, some RSV, so pan-RSV, and then A and B specific targets, as well as adenovirus, enterovirus, and human control RNase-P. So our first question we wanted to know is how the RPP assay performs with air samples, and how does it compare to Carmen and quantitative PCR results? So we have a common pathogen that we typically look at on these air samples, SARS-CoV-2, and that's present for all 3 of these assays. So we took 40 air samples from the 2023-24 school year and performed these 3 assays on those samples. The SARS-CoV-2 N gene is a common target for all 3 assays. RPP and qPCR both have the N1 and N2 targets. And Carmen, we just have one N gene target. I have tested other ones, but the one that we're using currently is the best performer. So one note for RPP and qPCR, sometimes only one target, N1 or N2, is positive. We think that these samples are actually positive for SARS-CoV-2, but maybe just at such a low level, or the virus is degraded enough that only one target is able to amplify. But we've gone back and forth of how we should call these samples. Should we call them positive or negative? Well, we can do two different analyses. One is a stringent analysis where for a positive result for RPP or qPCR, both N1 and N2 targets must be positive. In that case, the results look a little bit like this, which I'm showing you in the heat map below, where the positive is blue and the negative is white. We performed a kappa concordance test to compare sample to sample, how many times the samples agree between either qPCR and RPP or qPCR and Carmen. We find that the concordance was fairly similar for both of these tests. qPCR and RPP had a kappa score of 0.41, which means a moderate level of agreement. Similarly, we see the same thing for qPCR and Carmen. However, when we perform a less stringent concordance analysis, in this case, if we call either N1 or N2 targets positive for RPP or qPCR, we see that there's a lot more positive results for both of those assays. When we performed the concordance analysis, we found that comparing qPCR to RPP, again, the agreement was very similar to what we saw with the stringent analysis, with a kappa score of about 0.4. However, because there's so many more positive samples now for qPCR with that less stringent analysis, when we compare qPCR to Carmen, the kappa score is 0.04. Overall, we concluded that the RPP and qPCR data agree far more frequently, regardless of the stringency of analysis for SARS-CoV-2. However, there's a lot of other targets in both the Carmen and the RPP assays. So next, we decided to compare those two assays with samples from the 2024-2025 school year. So the samples we used are from 9 schools that have participated in the air sampling program since the beginning of the school year. So these samples were collected from August 2024 to, at the time of this talk, present day, which was March 2025. We have data for April now as well. For each sampling time period, which is about that 3 to 5 day window, we calculated the percent of cartridges that were positive for each target for each assay. And when we do that, the data looks a little bit like this. And there's a lot of stuff going on this graph, so I'm going to walk you through it. So we have our sampling date range from August until March. And then the Carmen assay results are in the heat map on the top. And the RPP assay results are in the heat map on the bottom. The darker blue indicates that there were a higher frequency of positive cartridges for that sampling time period. And the lighter color down to white indicates that there was a lower frequency of positive cartridges for that sampling time period. The overall trends that we observed, and I'll get more into these onto the next slides, was that the trends were similar between the two assays for several targets. Additionally, we noticed that the RPP assay in general was able to identify a higher higher frequency of percent positive samples for individual sampling time periods compared to the Carmen. I'll point out results here showed for pan-flu A. The Carmen assay does pick up pan-flu A during the January to March sampling time periods. The RPP assay picks up the pan-flu A target in that same sampling time period, but you can see that there's darker colors indicating that more of those cartridges were picked up to be positive via the RPP assay. Now to kind of get into looking at some of the individual comparisons. So here's Carmen and RPP comparisons for SARS-CoV-2. There wasn't much as far as trends go for the air sampling data here because SARS-CoV-2 has kind of it was sort of consistently positive so far throughout the school year. The RPP assay is shown, the N1 and N2 targets are shown as the black lines, and the carbon assay is shown as the green line here. Where things were pretty interesting is the trends for carbon and RPP for some of the other targets. So I'm showing on the top line seasonal coronaviruses, NL63, HKU1, and OC43. You can see for NL63 in particular, it was really cool to see that the trends for both of those assays were the same for NL63. We also picked up a little bit of HKU1 human coronavirus, and where things seem to be improved when using the RPP assay is some of these other targets. So we did notice that human coronavirus OC43, we were able to pick up an increased frequency of cartridges via the RPP assay that were positive for that particular target compared to the Carmen. We see that same trend happening where more cartridges are positive for the RPP assay compared to Carmen for flu A, flu B, and RSV. So our overall conclusions were that the Carmen and the RPP assays detect similar pathogens from air samples during the similar time periods in the 2024-25 school year. In some cases, as I showed you with RSV and flu B and flu A, RPP seems to have improved detection from the air samples. There's also room for expansion of the RPP panel that will allow us to detect an increased number of pathogens going forward. As far as our future directions, we do plan in the 25-26 school year to move to this assay exclusively for air sampling. We would like to, for some of our other ongoing studies, add the H5N1 target to the assay. So we can collect air sample data and potentially surveil for H5N1 as well. We'd like to add other additional seasonal respiratory viruses, such as parainfluenza viruses 1 through 4, as well as rhinovirus. There are rooms for improvement with this assay. We have noticed that there is, with H1N1 specific targets, there seems to be a mutation in the circulating virus that the RPP assay is unable to detect it. So we can improve this by adding additional primers and probes to the assay to improve detection. With that, I'd like to acknowledge members of Shelby O'Connor's lab that have been working on the K-12 project with me, particularly my undergrads, Jacob Mueller and Olivia Duvall, who've helped with a lot of the sample processing. I'd like to acknowledge members of Dave O'Connor's lab also working on the K-12 project, in particular Miranda Staus. She's been really super awesome of keeping these samples organized each week and keeping me mostly sane. With that, also, I'd like to thank the Public Health of Madison and Dane County, as well as our air sampling partners, the Dane County School Districts and the Orchards Program. I'd like to thank Brad Meyer and Luke Stewart from Standard BioTools for introducing us to this assay and providing reagents and training to use it with our air samples, as well as our sources of funding. All right, and with that, I'll take any questions.
Okay. Brad, did you want to pop back on for an update here?
Yeah, so it looks like if we kind of go maybe 2 slides down, yeah. Thank you, Dr. Ellis. That was fantastic and great work. Before we kind of kick off into the big Q&A session, I'd like to kind of just also put this out there. We noticed in the list of registrants that there were a lot of people from various public health labs. Some of you we know, some of you we don't. And so for those who are attending the APHL conference next week in Portland, we want to just kind of let you know that both myself and Dr. Ellis will be there presenting posters. Dr. Ellis will be presenting the poster on this research, as well as I will be presenting a poster on the RPP custom panel that Amy was using, as well as a few additional targets that I've been able to add, as well as we have a few other public health labs that do have our instrument that will be presenting. And so you can see that information here, as well as we have a user group meeting. That is on Tuesday, May 6th from noon to 1 p.m. where lunch will be provided. There's a QR code right there. Don't let the little dinosaur in the middle of it make you think that it's not active. It is active. And that's where you can register for the user group meeting. Space is limited, so it's best that you try to register as soon as possible. So that way we can make sure that there's room for you to attend. And we hope to see some of you over there. And feel free, if you can't make the actual user group meeting, please stop by our booth. Our booth is 319, and we'll be glad to answer any questions that you have.
Thank you, Brad. And I want to thank everyone for their informative presentations. We will now begin the live Q&A portion of the webinar. If you have questions you'd like to ask our speakers, please do so now. You can type them into the Ask a Question box and click Submit. Let's go ahead and get started. Our first question is for Dr. Ellis. It's asking, are the pathogens detected in the air by the assays you describe in your presentation also observed in the community in individuals? You're muted, Dr. Ellis. I'm so sorry.
Sorry about that. So 7 of the 9 schools that I showed you for the 2024-25 school year, 7 of the 9 of those schools are participating in nasal swab collection program. And so I do have a slide that I can show. They gave me permission to show this. So the nasal swab data, this was also performed with the RPP assay. So the targets are identical to the air samples for that particular assay. We found that I'm going to highlight one example. In February of 2025, we observed influenza B in those nasal swabs. And that was the first positive nasal swab was February 14th. When we look at the RPP assay influenza B results down here on the bottom, we observed that the first flu B positive air samples were around February 3rd. So the RPP assay can somewhat predict the incidence of viral pathogens detected in nasal swabs in schools.
Wonderful. Thank you, Dr. Ellis. All right, our next question here says, it looks like you used probe-based qPCR in both studies. Was dye-based qPCR ever considered, and what factors influenced your decision to go with the probe-based approach?
Hi, so for my study, it was more of an adaptation from what was already set up in clinical use. So we didn't consider dye-based. Although after the study had started and we'd already decided on probe-based, I did read about the CAS-based method, and it looked quite interesting. But we didn't end up going down that route because it would have been a lot of development for us, rather than just updating and moving what we had to microfluidics, which was more of the goal.
Okay, thank you.
All of ours have been pre-developed assays as well that we've adapted for air samples. So, and those have all been probe-based.
Excellent. Thank you both. All right, it looks like our next question is for Thomas. It asks, is nasal swabbing a reliable method, and would it be possible to exclude the DNA extraction process and use the swabs directly?
I think it's the best sampling method we've got for respiratory viruses, and it does vary depending on the virus and whether it's a predominantly upper respiratory virus or a low respiratory virus, and the stage at which the virus is within the host. So how many days incubation it's had, whether they are symptomatic or not. So but there's no reason why you can't detect virus directly from the swab. You will just lose sensitivity in most cases. And if you're looking at bacteria as well, that's something that you will definitely need extraction for. So you can swap the targets out of these assays. So it doesn't have to just be viral targets. If you want to use, you could also have host targets within that assay if you want to customize it to host targets, or bacterial targets, fungal targets. ITS2 and 16S would generally just tell you if there is a bacteria or fungi within that sample. But they definitely need extraction due to being much more robust than a virus. But yeah, even viruses you'd struggle to detect, I think, a sensitivity that was useful for a study generally. So extraction is pretty key. But extraction techniques vary, and you can have extractions that are much more simplified than others. Generally, viral extraction techniques quite straightforward in comparison to bacteria, where you've got a lot of lysis steps. I hope that's informative enough.
Yes, I think so. Wonderful, thank you. So we're going to try to squeeze in a couple more questions here before we end. Our next question asks, do you think air sampling can be adapted to sample exhale breath directly?
So we actually have done some of that. We've placed air samplers in household with individuals with known infections, and we do know that we can detect pathogens in individuals with known infections. So yes, the answer to that is definitely yes.
Wonderful, thank you. And we have time for one more question here. This last question asks, would you change out assay targets in the panel based on seasonality, or is it better to include all targets regardless of seasonality?
Hi, so I think, as I mentioned, seasons in our presentation, it probably depends on your study, but I'd be tempted to keep them in throughout an extended season, I'd say, probably. So, because seasonal viruses can shift based on the seasons being slightly different. And it depends on the seasons in which the country you're in. So you will notice a shift in viral patterns year on year, but the weather's not exactly the same each year. The seasons aren't the same each year. So there's going to be movement. But if your virus is particularly isolated to a certain season, then yes, you could. But you've got to account for a little bit of leeway either side, and potentially match it up with a primer probe target or a target that you're going to replace it with that matches the opposing season. Otherwise, you're just sort of wasting the space on the chip anyway. You might as well leave it on. And primers and probes don't tend to cost that much in the grand scheme of things.
Yep, I'd say the same for the air sampling too. Particularly as we're expanding to more global sites, it'll be interesting to see how the respiratory pathogens vary for northern versus southern hemispheres. As well as we've definitely gotten a few surprises with air sampling where things that are termed seasonal for the northern hemisphere have popped up like, oh, I wouldn't have expected this in April. So definitely there's a lot more to learn about some of these viruses too.
Excellent. I want to thank you both again for your time today and your important research. We would also like to thank Labroots and our sponsor, Standard BioTools, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. I want to remind all of you that you will see a survey pop up in a moment, and we appreciate your participation in this survey. This webcast can be viewed on demand. Labroots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.